Our vision, the goal of our research, is not only to understand the process of the formation of neurons, but to be able to recreate this process in situations in which it no longer takes place, for instance, after a brain injury.
Magdalena Götz has done much to increase the understanding of neuronal processes. As a university professor in Munich, she's dedicated her research to brain injuries. And as the director of the Institute for Stem Cell Research at the Helmholtz-Zentrum München, she examines stem cells in the developing and the adult brain. Together with her teams, she's made discoveries that have changed our knowledge of the brain. It's understood that neurons are irreplaceable. If they're destroyed by accident or by disease, they're irretrievably lost. Götz, however, found that neurons can be regenerated by stem cells in the brain. These stem cells, the glial cells, are unique. For nearly a century, their true potential was underestimated. Up till now, it was believed that the glial cell's sole task was to provide maintenance, support, and protection for neurons. Götz now discovered that the original glia, the radial glial cells located in the embryonic brain, are actually neural stem cells. The radial glial cells are predominantly found in the embryonic brain. They divide in different ways to generate neurons directly or indirectly and also self-renew. Götz made another discovery that shook the scientific world. She found out that after brain injury, adult glial cells, namely already differentiated astrocytes, have the potential to de-differentiate into stem cells. By means of electrical voltage, it can be shown that new neurons can be created from glial cells with the help of proteins. During contact with other neurons, only these new neurons can respond with an action potential. In other words, with the transmission of the stimulus along its axon. This cell transformation inspires hope among researchers.
We want to stimulate glial cells to make neurons, especially in the adult brain, by activating mechanisms which we initially identified as being important for brain development. In order to achieve this, we introduce regulatory proteins into the differentiated cells of the adult brain to regulate target genes, which in turn will re-enable the formation of neurons.
But there is still a lot of research necessary to selectively activate these neurogenic factors without any viral carriers, which currently still need to be injected into the brain. In addition, the functional integration of these newly transformed neurons into the tissue needs to be further investigated. There is still a long way to go before these findings can be implemented in a therapy.
We all hope that our results can soon be used in therapeutic applications, but unfortunately it's not possible to see into the future, and sometimes, even though one is very close to finding a way to implement the results, it can still take a long time. So we need to pursue several options in a broad approach to hopefully find a method as soon as possible.